Hello, Science 10, and welcome to Gram staining, part of lesson 2 on technologies that help us see a cell. So today, we are going to focus on a couple of concepts moving forward. So what you are trying to learn is what organisms does Gram staining work on? What structure does the stain affect? And what is essentially is the difference between Gram positive and Gram negative? In your notes, you're going to be answering questions based off of the information I give you as we go through this video. So let's start with the inventor extraordinaire, Hans Christian Gram. He invented this technique of staining different types of bacteria. Okay, his method has helped classify as many as 30,000 different types of bacteria. So he started um, up with this idea that helps us see the shape of bacteria. Because you have to remember, microscopes help us magnify things, but not everything has a color. Okay, some things are clear, some things are harder to see. So you have to give it a color to see it. And that is what he's doing with bacteria. So by staining them, all of a sudden bacteria are, which are super, super, super tiny single-celled organisms, living organisms, you now all of a sudden can see them under the microscope because they are a different color. So when we talk about bacteria, and I'm looking for them under the microscope, we can classify them into two categories. You are either purple, and that makes you gram positive, or you are pink, which means you are gram negative. So if you look at the picture I have over here, we can see gram positive bacteria and gram negative bacteria. Essentially what that means is they have two different types of cell walls. Now, we don't know what a cell wall is yet, but essentially it is a structure that protects our membrane, the cell membrane. Every cell has a membrane that helps transport nutrients and proteins and ions and really, really important things into the cell. Some organisms like bacteria and plant cells have this extra structure called a cell wall, which is an extra protection. Okay. If you are a gram positive bacteria, if you stain purple, you only have one space in between your cell membrane and the organism. And it's a peptidoglycan cell wall with lipoteatoic acids into it, surface proteins, teichoic acid as well. Okay, but there's only one extra level, cell membrane and then cell wall. A gram negative, okay, a gram negative, a pink one, has two cell walls or two outer membranes protecting its cell membrane. So it has the peptidoglycan, okay, it has lipoproteins, and then it has a lipopolysaccharide on the outside. So it has two distinct layers, okay, that protect that cell membrane. So a purple gram positive bacteria has one cell wall, and a pink gram negative bacteria has two cell walls. Okay. And this is a really important structure, and we'll talk about it more when we focus on the organelles inside the cell. So, like I said, some bacteria have one wall for their inner membranes and they take up the purple dye, and the other bacteria have more than one layer for their cell walls and they will pick up the pink dye. Again, one wall, purple, two walls, pink. So this is the process in which we have to stain bacteria in order to find out if they're gram positive or gram negative. So you have this drawing in your notes and you need to label each stage as we talk about it. So the first stage is fixation. Okay, so getting them prepped and ready for the dye to uptake. Okay, then the next thing you do is you dye them with something called crystal violet. All right, crystal violet turns them a very dark, dark, dark purple. And then what happens is you give them an iodine treatment. And that iodine treatment, okay, essentially helps keep that stain in. Okay, it helps set that stain into the cell wall, okay, because we're staining the cell wall. But if you are gram positive, when you put the decolorization onto it, it'll stay purple. If you have two cell walls, okay, it will actually remove the stain. So what you're doing here is crystal violet is the dye, iodine is treating it. So if we go back to the slides over here, what we're looking at is the dye is trying to set, the purple dye is setting into this cell wall here, okay? When you dye a gram negative bacteria, it's setting into this first cell wall and it's not actually being taken in because the structures in this one are the ones that the purple dye adheres to. So when you go through the process of decolorization and you add something to remove the dye, because gram positives are held onto that the dye by that cell wall, it'll stay purple. But this one, the gram negatives, will actually turn back to the original color because they don't have that cell wall that likes to hold on to that purple dye. It's not accessible because there's a second wall. So what you do is you counter stain with safranin, which is another type of dye. And when you do that, okay, the second type of bacteria, the gram-negative bacteria, its outer wall really likes to take up the pink dye. So now you will have a pink-stained bacteria. So these types are you're able to look at under the microscope and see either purple or pink, okay? Now, this helps classify them into the gram-positive and gram-negative categories, which is important because that's a very big category, okay? Um, and it has a lot of different types of bacteria that are part of it. So like Staphylococcus, Streptococcus, Staphylococcus epidermis, these are all just different types of gram-positive bacteria. Some of them, Staphylococcus aureus, for example, is an infection that's really dangerous, and it's like you can see it, and it causes skin infections. It can cause pneumonia and heart valve infections. Where if you have a gram-negative bacteria, those are different, and you can start looking at those as different ones. So gram-negative bacteria involve things like E. coli, Pseudomonas aeruginosa, Calbelsa pneumoniae. Okay, these are our very fancy names for them. Okay, essentially, most of these are just different types of infections. Okay, Pseudomonas aeruginosa is a germ that causes infections in humans, usually people in the hospital. It can cause pneumonia or infection of the blood, and it's really dangerous. Okay, so when we look under this microscope, we can actually see two different types of bacteria. So if I was to look under a microscope, I might see something like this. You can see now that there are gram-positive and gram-negative stained bacteria. The gram-positive are circular in shape, and the gram-negative are more rod and cylinder shaped. Right? So if I was looking underneath the microscope at a scraping of something or a sample of something, before I would look under, I would stain it so that I could see all of these different types of bacteria because I might not see them without the stain. Why is this not? There we go. So here's another example of gram-positive and gram-negative. You can just see the different colors that they are, okay? And ladies and gentlemen, that is essentially gram staining. And why we use it is because it helps us identify different types of cells, single-celled organisms such as bacteria, okay? So what I would like you to do is make sure that you have answered the 5 questions in your notes and added the information on top of your picture that I gave you. And we will talk to you